We will start now the remaining properties of the enzyme, then the different types of the enzyme and the classification of enzyme. Yesterday in last lecture we studied very well, that is different characteristic of the enzyme, and today we start from this remaining point. So students, enzymes commonly enjoy one or more of the following strategy to catalyze the specific chemical reactions in the body. Covalent catalysis. The process of covalent catalysis involves the formation of covalent bond between the enzyme and substrate. The modified enzyme then act as a reactant. After the completion of the enzymes return to its original unmodified state, and the proteolytic enzyme chymotrypsin provides an excellent example of this mechanism. Then next one, metal ion catalysis. The metal ion may be bind to substrate and increasing the number of interactions with the enzyme and thus binding with that substrate. Then general acid base catalysis. In general, acid base catalysis, molecules other than water plays a very important role in proton donor or acceptor. Then reversibility of reaction. The enzymes are capable of bringing about reversion in a chemical reaction. The digestive enzyme catalyze the hydrolytic reactions which are reversible. Isozymes. Certain enzymes occur in multimolecular forms in the same organism. These enzymes have the different physical properties but catalyze the same type of reactions. For example, lactic dehydrogenase is an example of isozyme which can be distinguished into 5 distinct types. Then next one, the zymogen. All inactive precursor of enzyme is called the zymogen, and zymogen is cleaved to form the active enzyme. Means zymogen are the inactive form of enzyme and which cleave to form the active enzyme. Then next one, allosteric nature of the enzyme. Allosteric enzymes are a class of regulatory enzymes which increase or decrease the catalytic actions. Then allosteric enzyme function through reversible non-covalent binding of regulatory compounds called modulators or effector. The modulators for allosteric enzyme may be inhibitory or stimulatory. Modulator is the substrate itself. Regulatory enzyme for which substrate and modulator are identical called the homotrophic enzyme, and when the substrate and modulator are different called the heterotrophic enzyme. Then how to nomenclature, how to designate it or given particular name to particular enzyme means what is the nomenclature of the enzyme and what are the rules behind it. So many enzymes have common names. For example, trypsin, a proteolytic enzyme secreted by the pancreas. So common name provide little information about the reaction that enzymes catalyze. Many enzymes are named for their substrate and for the reaction that they catalyze with the suffix ase added. For example, protease means the enzyme which brings about the action on the proteins, proteolytic enzyme. Pancrease, which derived from the pancreas. Understand? So ATPase means these enzymes are very important for the synthesis of ATP. Because of the confusion that arose from these chemical names, common names, International Commission of an Enzyme was established to create the systematic basis for the enzyme nomenclature. Enzyme Commission have developed a rule for naming the particular enzyme. According to this rule, each enzyme is classified and named according to the type of chemical reaction it catalyzes. The Enzyme Commission has given each enzyme a number with 4 parts. Then now start the classification of enzymes. So the first one, the International Enzyme Commission has classified all the enzyme in following 6 classes. So first one, oxidoreductase. So this name indicate their meaning. Oxidoreductase means this type of enzyme brings about the oxidation and reduction process, which brings about the oxidation and reduction process. Oxidase reductase catalyzes the oxidation reduction reaction, transfer of electrons. Then dehydrogenase. catalyze the removal of two atoms of hydrogen. Oxidases, this catalyze the reduction of O2. Oxygenase, catalyze the incorporation of molecular O2 with the substrate. Peroxidases, catalyze the hydrogen peroxide as the oxidant. So these are the different examples of the oxidoreductase type of the enzyme. Then transferase. Transferase are the catalytic reaction, transferase, that brings about the catalytic action that involve the transfer of groups from one molecule to another. Examples of such group include amino, carboxyl, carbonyl. Common names of the transferase which brings about the means include the general prefix trans. Okay. Then hydrolases. Hydrolases catalyze reaction in which cleavage of the bond is achieved by adding water. Different examples such as peptidase, glycosidase, esterase, phosphatases, deaminases, and so on. Lyase catalyze a reaction in which group are removed to from double bond or added to a double bond. Then isomerases. This is a heterogeneous group of enzyme. Isomerase catalyze several types of intramolecular rearrangement. Then ligases catalyze the formation of bond between the carbon and nitrogen. Then what are the different factors affecting the enzyme activity? So I already explained in last period ki generally pH, temperature, then time, then effects of the enzyme concentration. Just in last period these are the different factors are listed, but now these factors are studied one by one. So first upon time. In an enzyme substrate reaction, mixture at a constant temperature, the reaction velocity decreases at the time progresses. Means when the time is increased, so reaction mixture means when we going from the proceeding one by one stage. So the concentration means mixture at a constant temperature, the reaction velocity decreases at the time progresses. Means the decline may occur for the number of reactions. The approach of an equilibrium and the associated influence of the reverse reaction means the change in pH if the medium is inadequately buffered also means pH, then temperature and the time having some correlation between them. Then next factor about the temperature. So the enzyme activity increases with increase in temperature, which reaches a maxima, and then the activity declines with further increase in temperature. Enzyme activity with temperature gives a typical bell-shaped curve on graph paper. The temperature at which the enzyme exhibits maximum activity is called as the optimum temperature. Each enzyme has an optimal temperature at which reaction rate is greatest. Without denaturing the enzyme, the temperature allows the greatest number of molecular collisions and the faster conversion of the reactants to product molecules. Then next factor about the pH. The hydrogen ion concentration has a marked effect on the charges on the ionizable group in a protein molecule. Such effect can alter the three-dimensional structure of protein. The structural alteration may alter the geometry of the active site. So varying degree and ionization of group, particularly to the active site, they have the remarkable effect on the enzyme substrate complex. What is meant by this enzyme substrate complex? I already explained in last period. The substrate, the enzyme react with the reactant on which it become act. That is the substrate. Typical bell-shaped curve here obtained. Then next effect of enzyme concentration. When substrates are used in large excess, the velocity of the reaction is proportional to concentration of enzyme. Velocity of the reaction is proportional to concentration of the enzyme. And when the substrate is in excess, so this enzyme is saturated with, and the velocity of the reaction is maximum. Means velocity is directly proportional to that the active site of the enzyme. Are you understand what I mean to say? If the enzyme reaction is double with the substrate, it still in the excess, the velocity should be double also. And this measurement of relative nature of the active site of the enzyme with respect to their velocity, it is very important for the effect of enzyme concentration. Then next one, effect of substrate concentration. Keeping enzyme concentration constant, so increase in substrate concentration, which results in a very rapid rise in velocity in the beginning. So if the substrate concentration is increased further, the velocity of the reaction remains practically constant. The maximum velocity attained is indicated by the Vmax, and the substrate concentration at half maximum velocity known as Michel-Mendon constant, which is denoted by Km. So this is very important. Then next property, redox potential. Many enzymes are sensitive to oxidizing and reducing agent, and the comparative ability of the oxidation or reduction of an enzyme is known as redox reaction. Means it brings about the two kind of process that at a time, that is oxidation and reduction, and it is a redox reaction, just like the process of photosynthesis, which is a redox reaction. Then next property about the enzyme inhibitors. Many types of the molecules of enzyme exist which are capable of interfering with the activity of the individual enzyme. And these some enzymes are normal body metabolites that inhibit a particular enzyme as a part of the normal metabolic control of the pathway. So students, these are the different type of enzymes, and they brings about the different type of action. So thank you very much. झालाय माझं